Dr. Shipley is getting her slides ready to share. I would like to introduce her. Before I do that, want to let you know that to save bandwidth for the presenters, we would prefer if you mute yourself and turn off your video. And if you have questions during the presentation, please put them in the chat box, and we will moderate those at the end of this session. So, Dr. Lisa Shipley is a professor of wildlife ecology in the School of Environment at Washington State University. She obtained a BS in wildlife biology at Colorado State University, an MS in wildlife management at University of Maine, and a PhD in wildlife and fisheries science at Texas A&M University. Her research interests include foraging behavior, nutritional ecology, and habitat ecology of wildlife, especially wild herbivores such as deer and lagomorphs. Her research focuses on both free-ranging and captive wildlife, which has resulted in over 100 journal articles and book chapters. For this work, she has collaborated with many scientists, agency biologists, her 44 graduate students, of which one is me and several others that I see have joined us today, and postdocs, and over 100 undergraduate researchers. Lisa has also conducted research internationally in Sweden, Australia, Argentina, and Slovakia. She established and currently directs the Wild Ungulate Facility and the Small Mammal Research Facility at WSU, what we affectionately call WOOF and SMURF, respectively. Between 2002 to 2011, she led the captive breeding of endangered Columbia Basin pygmy rabbits at WSU and assists in the ongoing restoration efforts in Eastern Washington. So at this point, please mute your audio and turn off your video, and I will turn it over to Lisa.
All right. Hopefully you can see and hear me. Any problems there? Okay.
You're good.
Okay, good. Well, I'm really excited to be able to give a webinar for you today. I have been a member of the Nutritional Ecology Working Group since it started, and it's especially happy for me that one of my former students could introduce me today. So we only have to look around to see how drastically human activities have altered wildlife habitats, both across the Pacific Northwest where I work, let alone the rest of the world. Now, if you look a little closer, you might also see land and wildlife managers hard at work restoring and improving habitat quality. Therefore, understanding the effects of both habitat alteration and restoration on wildlife populations is crucial, especially in the era of climate change. Now, for wild herbivores, many of the population effects of habitat change are expected to be mediated through their effects on the quality and quantity of forage, how forage influences the nutrient animals can acquire, and how those ingested nutrients are converted to body stores and reproduction. And survival and reproduction of individuals in turn drives population dynamics. So today I'd like to talk about a 20-some year journey that I have been through that aimed at exploring each of these links in 3 native deer species/subspecies in Washington. Now I'm going to be a little unconventional and start with the far end of this flowchart and work my way backwards. And I'm doing that because to determine what metrics are best to assess habitat disturbance and restoration, we first need to demonstrate that there's a relationship between food consumed by deer and their individual fitness. Although several studies have linked body condition to fitness in wild herbivores, far fewer have been able to demonstrate this causal link between the amount of digestible energy consumed and fitness because it really requires intensive experiments with captive animals. So therefore, our first project was to conduct a large manipulative feeding experiment at the Wild Ungulate Facility at Washington State University, in which we could measure daily digestible intake and measures of reproduction and body condition. And we exposed 24 lactating does with fawns to 1 or 3 nutritional treatments, and we repeated that over 2 years. Animals were individually fed, so we could measure their daily digestible energy intake across summer and fall. Now, each treatment group in each year received 1 of 3 pelleted diets that mimicked the digestible energy content of forages as they declined from late July through early fall. And those ranges were typical of those that are found in natural habitats in eastern Washington. In October, we bred the females and assessed their pregnancy with fecal progesterone and ultrasound. After fawns were born, we measured milk quality and nursing behavior, and fawn growth and mortality, and we measured female body condition periodically throughout. And just going to give you a brief highlight of what we found that's relevant to the talk today. We found that mule deer that consumed more digestible energy before the breeding season were more likely to become pregnant, give birth to more twins than singles, had higher protein milk, and faster growing and longer living fawns. They also had more stored fat going into winter. And yes, in case you're wondering, throughout these slides, you might see DEI, and I'm actually referring to the other DEI, digestible energy intake. Now, as for establishing the link between digestible energy intake and fitness, we'll next examine this intermediate link between forage characteristics and digestible energy intake of wild herbivores. And this has been the focus of our most recent research, therefore will be the largest portion of this presentation. And establishing this link and determining which forage metrics best predict DEI of wild herbivores has remained challenging for many reasons. First, digestible energy intake is normally estimated as the product of bite size, bite rate, time spent foraging, and the digestible energy content of the diet. Each of these components are difficult to measure in wild herbivores, and small measurement errors in each of these can compound when these four components are multiplied together. Second, each of the four components of digestible energy intake is influenced by a variety of environmental and animal characteristics, often in complex and inverse ways. Many of the known mechanical relationships between these variables are curvilinear and as indicated by the equations in the white boxes. Furthermore, there is a volitional component to intake whereby animals can voluntarily adjust their behavior, such as how fast they want to walk, which bites to choose, to compensate for forage conditions. Now, a final challenge has been to figure out how best to measure the characteristics of forage resources that actually drive digestible energy intake. So therefore, we compared the ability of 7 potential metrics of forage resources to predict digestible energy intake and its components for deer in semi-wild conditions. Now, each of these metrics represents a measure of forage resources that we would expect to influence either forage intake or forage quality. So the simplest metric is total forage biomass. And the gold standard for estimating plant biomass is clipping all the vegetation on microplot plots within the relevant study sites, sorting by species and plant part, drying at 100 degrees Celsius and weighing it. And this is the approach that we use for the work that I'm going to present today. However, as many of you know, sampling biomass this way is very time intensive. So time can be saved by using ocular estimates of plant cover on plots. Using those cover estimates, however, requires first developing accurate cover to biomass conversion equations from relatively large and well-distributed sample sizes and accounting for covariates such as phenology and overstory canopy cover. And if you're curious about how that might work, feel free to check out a recent paper from our lab group by Monzingo et al. 2022. Now, the next 3 metrics, top species, accepted, and selected biomass, account not only for plant biomass, but also indirectly for forage quality. By restricting the forage biomass that we're going to count or consider available to only plants that the animal consumes or selects. Therefore, it requires additional measurements of diet composition and or selection. Now, top species biomass, we define that as the biomass of plants species that forms at least 1% of the diet for that animal species, location, and season. Now, fecal analysis has been the go-to method for determining diet composition for wild herbivores for quite a while. However, both the older microhistology and the newer DNA metabarcoding methods often fail to accurately identify and quantify plants consumed. The more accurate bite count method can be impractical in many situations because it requires habituated animals. For the work I'm presenting today, we did have those animals to work with. Now, estimating accepted biomass, which is the biomass of plant species that are used in equal or greater proportion to availability, and selected biomass, where use exceeds availability, requires going even one step further. A selection index must first be developed for each plant or plant part from the percent used, which is diet composition, and percent of plant available, and these need to be measured in the same location and season. Now, for the next two metrics, suitable biomass and community digestible energy, the nutritional quality of plants is measured directly instead of being inferred from animal choices. Now, measuring digestible energy content of forages requires collecting fresh samples and analyzing them for gross energy, sequential neutral detergent fiber, and in many cases, tannin binding capacity. Then digestible energy and digestible protein, if you need that too, is estimated from those values using summative equations developed for deer and elk by Robbins. Also, there's equations for moose developed by Spallinger, and I believe there's a black bear model as well. Now, for community digestible energy, the digestible energy content of plants is weighted by their relative abundance in the habitat, determined through this biomass sampling. Now, suitable biomass is a little more complicated. It's the minimum biomass of forage that together meets digestible energy and digestible protein requirements for that animal species. This metric is calculated from biomass and the digestible energy and protein content of available plants. Now, this method also requires a good estimate of the requirements of your animal species for dietary DE, dietary digestible energy, at the appropriate reproductive status, such as lactation or maintenance. Diet digestible energy requirements really can only be determined through controlled captive experiments, and these are available for some wild herbivores, such as deer and elk, but not all. And we're especially lacking information for wild bovids and pronghorn. And because digestible energy and digestible protein can vary with animal size and digestive system, the model works better if you have species-specific requirement data. Now, suitable biomass is then estimated from the fresh deer model using linear programming that figures out how much of the biomass on average is going to meet those requirements. Now, the final metric is community bite size, and that's based on the strong and well-known positive relationship between the size of bites animals take from their forage and harvest rate in grams per minute. If you might recall, that was one of the components of digestible energy intake. Now, average bite size taken per plant is most accurately measured, again, from bite count methods, but could potentially be acquired from video collars or browsed twig diameters. For the latter, that would have to be just looking at senescent hardwood plants, might work for moose in the winter. So to estimate community bite size, average bite size per plant species is weighted, again, by their relative abundance. So to test how well each of these 7 metrics of forage resources predicts digestible energy intake and its components, we leveraged a large data set of 153 sites across widely varying study areas in Washington. And each of these sites, we had collected concurrent data on both forage resources and digestible energy intake of deer. We collected nearly 1,000 diet and forage samples from the 3 deer species and subspecies in the Odocoileus genus: mule deer, black-tailed deer, and white-tailed deer. Now, our 3 study areas included Western Washington, which will be shown in blue throughout the rest of this presentation, Northeastern Washington, which will be in green, and Southeastern Washington, which will be in pink. And these study areas spanned 5 ecoregions and 5 vegetation zones and varied widely in precipitation and temperatures. Now, the Western study area was on the west side of the Cascade Mountain Range, so very wet and productive. And the Northeast and Southeast sites were in the rain shadow on the east side of the Cascades, So we're either dry forest in the north or dry grassland in the south. But even sites within each of these study areas varied manyfold in plant biomass, local environmental conditions. So this variety allowed us to explore the universality of forage metrics for deer. So we've already talked about how to measure forage metrics, but how we measure digestible energy intake at each site. We had to start with tractable deer that we'd hand raised at the wild ungulate facility. And as they grew, we exposed them to native forages and trained them to experimental protocols. So as yearlings and adults, we transported 4 to 8 deer per site to fenced half-hectare or 1-acre enclosures at each of those 153 sites across Washington. At each site, we recorded the species and size of each bite taken by each deer within several foraging bouts per animal per day. We then collected simulated bites for each plant species consumed so that we could then estimate bite size and to create simulated diets each day with that observed plant species composition. Now we determined the digestible energy content and digestible protein of simulated diets in the lab using the same methods as for the forages that I described previously. We measured time spent foraging using activity sensors on the collar of the deer and calibrated these with direct observations. So we first compared forage resources and foraging variables across the 3 study sites, then evaluated models predicting digestible energy intake and its components from each of those 7 forage metrics. And we used different model forms, a linear, power function, and Michaelis-Menten form. Michaelis-Menten is a asymptotic relationship that we often find when we look at functional response of herbivores. And then we used AIC to rank the models. Now, before I start on results, you're going to see several tables and just a little heads up on my coding system. The green highlights are the highest means. Yellow would be the second highest means. And later on, you'll see some orange, and I'll explain what that is later. So forage resources varied up to 8-fold among our study sites, with the Western study area shown on the left producing the most total accepted and selected biomass. The Northeastern study site had the most suitable biomass, and the Southeast area had the greatest community, digestible energy, and bite size. And you'll note that top species biomass was essentially equivalent across study areas. Now, the composition of plant functional groups available also differed, as you might expect, across the study areas, with the highest relative abundance of conifer, forbs, and ferns, highest in the Western study area, evergreen and deciduous shrubs in the Northeast, and grass in the Southeast. Now, although Western Washington had over 3 times more total forage biomass, deer actually had a higher bite rate, diet quality, and digestible energy intake in the Northeastern and Southeastern study areas. Okay, so let's cut to the chase now. As for which metric best predicted DEI across those study areas, suitable biomass, which again, remember, combined both biomass and DE content of forbs, and that's shown in green here, was the best metric to predict DEI, and it explained 21% of its variation. If we added study area as a covariate, the R-squared increased to 0.33. Now, top species biomass, that included only the biomass that composed greater than 1% of the diet, is shown in yellow. It was second best at predicting digestible energy intake. And total accepted and selected biomass in orange all performed better than the null model down at the bottom, which is highlighted in gray. However, for those 3, the R-squared was only 3%. Now, the relationship between suitable biomass and DEI for each study area were fairly similar. You can note the overlap in the different colored points. We found that across study areas, deer required at least 400 kilograms per hectare of suitable biomass to meet their digestible energy requirements for summer maintenance. And that is indicated by that horizontal orange line at 15,300 kilojoules per day. And those requirements were only met in 27% of the sites sampled. Now, to dig a little further, we next examined which forest metrics best predicted the components of DEI, starting with harvest rate, which is a product of bite size and bite rate. Now, although harvest rate responded to all biomass metrics, top species biomass was the best model, and the best form was a power function. And then the next best model was community bite size. Now, the relationship between harvest rate and top species biomass in our study is indicated by that black asymptotic line. And what's really interesting is it exactly matched the relationship from an earlier study that measured intake a different way from esophagically fistulated mule deer. And this was also in eastern Washington. And you can kind of only barely see that little orange line underneath our black line. Top species biomass, however, only influenced harvest rate until it reached about 200 kilograms per hectare, at which point it leveled off. So we next modeled time spent foraging per day. Now, across study areas, time spent foraging per day was influenced by the community digestible energy, followed by community bite size, and then suitable biomass. These were the only three that performed better than the null model. However, those study-wide relationships between foraging time and either community digestible energy on the left or suitable biomass on the right were really driven by the Southeast Study Area site, which is the pink and circled, because that site had relatively low foraging time coupled with a high community digestible energy and low suitable biomass. And so when sites were analyzed separately, this relation disappears. And so we believe that was just related to differences among study areas rather than some overall mechanistic relationship. So finally, we examined how diet quality responded to these forage metrics. The digestible energy content of the diet was best predicted by community digestible energy, followed by suitable biomass, but total, accepted, and selected all performed better than the null model. The relationship between dietary digestible energy and community digestible energy was most pronounced in the Western study area. Across the study areas, deer met their maintenance requirement for dietary digestible energy, which is indicated by, again, that horizontal orange line at 11.5 kilojoules per gram, in 43% of the sites, mostly in the Northeast and Southeast study sites. Okay, so when selecting a metric to measure characteristics of forage that will best predict the effects of habitat changes on population performance, our analysis suggested that first, the relationship between digestible energy intake and forage resources was relatively similar across the study area and deer species, so these results were relatively robust. Second, suitable biomass was the best overall predictor, but it does require that you have information about the nutritional quality of the foods. That, of course, is a very costly analysis. So our results and my suggestion is that the nutrition and wildlife communities help develop an open access database repository for the forage quality data. And I know that Mark Edwards talked in a previous NUG seminar, and he, I think, presented, I wasn't able to go, I had another meeting, but talked about some efforts to try to collect those data and develop that for wild herbivores. So hopefully those of you who have those data sets will participate. In addition, we need investment in more basic studies of nutritional requirements that we need to calculate suitable biomass, and especially for wild bovids and pronghorns. In my career, I found that these are the most difficult projects to get funded. They're not theoretical enough for NSF and not applied enough for state and federal management agencies. So maybe we can get our heads together and figure out how to support those. The second best predictor, top species biomass, does not require nutritional analysis, but does require accurate and precise measures of diet composition. And so investment in further developing DNA metabarcoding or sequencing techniques for diet composition is what I consider one of the most urgent things that we need in herbivore ecology. I'm very excited, though, about some new developments by Tal Levy and his postdoc Charlotte Erickson at Oregon State University that should be coming out soon. And hopefully they'll be able to present to you maybe next year. So choosing which forage metric to use is really going to depend on what data are most available for your study system, nutritional analysis or diet composition. However, even the best forage metric in our study explained less than 25% of the variation in DEI. Therefore, we also need to continue to develop better understanding of animal behavior and plant chemistry and plant structure and how all of those things drive DEI. This is another research frontier, I believe, in nutritional ecology. Okay, so now that I've demonstrated that suitable biomass is the best metric for predicting DEI in our study with deer, I will end with 3 case studies that examined how land management activities influenced forage resources, in particular suitable biomass for deer. These include the effect of herbicides and even harvest on black-tailed deer in the West Coastal Cascade Forest in Western Washington, forest thinning and fuel reduction treatments in the dry forest of northeastern Washington, and cattle grazing on mule deer in the dry grasslands in the southeast. So let's start with our first case study. We examined the effects of herbicides and stand age created by even age forage management on black-tailed deer across much of Western Washington. On industrial or commercial forests, herbicides have largely replaced prescribed fire as a site preparation technique because fire violates air pollution guidelines. They are often applied prior to planting seedlings and sometimes once after the first growing season. Now, the herbicides are intended to reduce competition between the young conifer seedlings and that extra vegetation, which happens to, a lot of it, be wild herbivore forage, to enhance the survival of these seedlings and growth and to promote stand progression. And this is most important in the highly productive areas. So herbicides are a key tool used by silviculturists for managing young forests, at least in Western Washington. Now, however, some biologists have been concerned that herbicides reduce the amount of forage for black-tailed deer and potentially Roosevelt elk and could change the composition, thus quality of forage species available. By speeding up the growth of conifers, herbivores might also truncate the window of time stands spend in that open canopy. So the 96 sites that we used in our study were distributed within the Douglas fir and western hemlock forests across much of Western Washington. And for those not familiar with Washington, the top arrow indicates Olympia, the capital, which is south of Seattle, and the lower arrow shows Portland, Oregon. The deer and the tree symbols indicate those samples the sites we sampled. Because forest stands take so long to develop, this was a retrospective study. And we worked with private timber companies and a few other groups to locate forest stands that were harvested at the same time, but some sections had been sprayed with herbicides and other sections were not sprayed. This allowed a split plot experimental design, which improves the power of statistical tests. We found sites with paired treatments across all stages of stand development and grouped them into 5 stand age categories: 1 to 3, 4 to 6, 7 to 9, 10 to 13, and 14 to 20. And then we included unmanaged min-zero stands, which were established before herbicides were used, and this was used as a comparison. So I'm only going to show one slide from that huge study, but this is the one that's relevant to what we're talking about today. So we found that suitable biomass for black-tailed deer was highest from 4 to 10 years post-harvest, and the peak was several hundred times higher than when stands reached the age of above 20 years, when the tree canopy then shielded the understory vegetation from sun. Now, spraying with herbicides, which is indicated by the yellow line, reduced suitable biomass compared to unsprayed pears, but primarily in the first years post-planting and post-herbicide application, although there's some hint that these effects could last further on in stand development. Okay, let's move on to the second case study. And in this one, we investigated the effects of thinning, fuel reduction on St. Patrick mule and white-tailed deer in northeastern Washington. This area is dominated by dry Douglas fir ponderosa pine forest that historically supported frequent low-severity fires that maintain these light fuel loads and low stocking densities of timber. However, over a century of fire suppression and some ill-suited forest management techniques have left large tracts of forest with heavy fuel loading, high stocking densities, and continuous canopy closure. Not only do these conditions increase the likelihood of unusually severe wildfires, but it also reduces the amount of light that penetrates to the forest floor, limiting the growth of herbaceous and woody understory vegetation that provides food for wild and domestic ungulates. Therefore, reducing fuel loads and restoring these historic conditions is a top priority of many public forests throughout the western United States. In Washington, the Collaborative Forest Landscape Restoration Program, or CFLRP, harnesses the skills and resources of federal, state, and private landowners to apply fuel reduction treatments such as thinning and burning. Now, by opening up that forest canopy, these treatments have the potential to improve forage resources and increase the nutritional value of this landscape for wild herbivores. So we tested this hypothesis within the Colville National Forest in northeastern Washington. The study area was bounded by Canada to the north and the Colville Reservation to the south. And over 5 years, we sampled 82 treated and 26 untreated forest stands that range from 2 to 100% canopy cover and 1 to 20 years post-treatment. Stands that we sampled were spread over a range of ecological conditions within this broad geographic area. Again, you get a one slide result here, but on average, the thin sites had a 50% reduced canopy cover and a 125% increase in suitable biomass. And the suitable biomass increased to a peak at 12 years post-treatment and decreased with canopy cover, which is indicated by the increasingly dark yellow lines. Okay, so our final case study examined the effects of cattle grazing on mule deer in southeastern Washington. State wildlife agencies that are charged with managing wildlife within their borders often seek to purchase large tracts of lands that represent important ecosystem and plant communities that then could be managed specifically for wildlife. In Washington, many of these lands are contained in old ranches, and the owners and communities agree to selling the lands if they could be maintained as a working landscape and continue to provide economic and cultural role in the community. Therefore, agencies are interested in whether continuing the historic livestock grazing is compatible with managing these habitats for non-game and game species such as deer. Now, some evidence suggests that cattle grazing might actually improve the nutritional quality of forages available to wild herbivores. First, cattle grazing might reduce the proportion of standing dead biomass in the bunch grasses that are in our area, which then may increase the accessibility of that higher quality green vegetation for the smaller herbivores. Second, grazing just before boot stage might increase the quality of grass by delaying its phenology and allowing the leaf tissue to cure at a younger, more nutritious stage. This is called the foraging conditioning hypothesis and is used by hay farmers to decide when to mow their hay. Finally, by reducing competition with grasses, grazing might promote more digestible forbs and shrubs that are preferred by a lot of the wild herbivores like mule deer. Now, on the other hand, if the diets of cattle and deer overlap substantially, forage removed by cattle then becomes unavailable to mule deer, at least for one season, and therefore might affect the number of deer that can be supported and the intake of individual mule deer because it can increase search time and reduce bite size. So this study took place in the Steep Canyon grasslands, a blue bunch wheatgrass community in southeastern Washington. We created grazed and ungrazed treatments using electric fencing to exclude cattle during spring grazing. The target was for the utilization by cattle of 40 to 60% of the bunch grass biomass. This design again allowed a split plot design with season and pasture. We had 3 different pastures, and for each we established 6 paired grazed and ungrazed plots of 1 acre, 3 for spring and 3 for fall sampling, which resulted in 36 total plots. Within a pasture, the paired plots were greater than 100 meters apart, and the grazed plots were adjacent to the ungrazed plots. So we found that suitable biomass did not differ between grazed and ungrazed pairs in spring right after grazing occurred, but 4 months later, as plants matured, the effect was more evident. Suitable biomass for deer in grazed areas was only half that of ungrazed areas. Now you might notice something that seems a bit counterintuitive, that the overall suitable biomass was way less in spring than fall. However, this is because in the fresh model that we use to calculate suitable biomass, you need to put in the maintenance requirement. And we used a higher maintenance requirement for mule deer during the spring when they're lactating. In the fall, when they're weaning, we used a maintenance requirement. So you can't really compare the absolute suitable biomass between the two, but you can look at the relative difference between grazed and ungrazed. In a follow-up study, we went back 2 years later and looked at suitable biomass and 2 years later, and that was a period of time where there was no grazing occurring, and the effects of grazing on suitable biomass for deer disappeared or was no longer evident. Okay, so we found from our case studies that suitable biomass was highest where tree canopy was lowest, whether by even age harvest or by thinning, and we would expect to find maybe similar things for a burning treatment. We also found that herbicides and livestock grazing can reduce the biomass or nutritional quality of plants, thus suitable biomass for wild herbivores. However, the responses of suitable biomass may or may not show up the same way they did in our case studies because they would be expected to vary with animal species, different sizes, different nutritional requirements, and diet choices with the size, intensity, and frequency of the habitat treatments and the location and season of these applications. So in conclusion, our 3 case studies demonstrated how suitable biomass can be used to evaluate the effects of common land management activities on the digestible energy intake of deer. And you can take this even further. For example, suitable biomass can be used to map foodscapes over landscapes, such as those that have been recently created by Ryan Long and his lab at University of Idaho for deer, elk, and bighorn sheep. So finally, what are the next steps in this very long project? Well, before I completely wrap up this work, I'd really like to save biologists a lot of time and money by training deer to maybe measure and manage their own habitat. And I'll be on sabbatical next year, so maybe, just maybe, that would be a great time to get started on that really difficult task. So finally, I'd just like to acknowledge my 2 co-authors on this, at least the intermediate part of this talk, Amy Ulaffa and Ivor Hull, and the many of my other former graduate students who collected the data that I presented today, including Sarah Wagner, Stephanie Berry, Troy Tollefson, and Deborah Monzingo. And this work was supported not only by WSU, but by a variety of agencies and organizations, such as the U.S. Forest Service, Washington Department of Fish and Wildlife, Nacassie, the Quinault and Muckleshoot tribes, and a number of timber companies. So with that, I'd be glad to take any questions.
All right, great. Thank you so much, Lisa. We have a question already. Karen Grant says, this is a peripheral question. There has been some interest in captive wildlife nutrition related to endangered species reproduction in some ungulates, where the body fat content of mother affects ratio of male to female offspring, and increased body fat skews that ratio towards males. I was wondering if you track gender of offspring based on DEI of the does. That sounds very familiar, doesn't it, Lisa?
Yes. Um, I'm trying to remember. I think the study that we talked about where we actually the captive study where we fed them different levels of nutrition, I don't think there was a male-female ratio that changed with differences in DEI. Um, we did also measure body fat, and I didn't present that so much today just because I was talking about digestible energy intake. But I do think Tamara might want to answer that because that was part of what she was working on for her PhD. Um, so,
Yeah, we didn't really see anything. We looked at it in elk as well, and we didn't really see that, Trivers-Willard effect of skewing towards males with the body fat that we expected.
but our sample sizes to do that kind of work, sample size is an issue because you have to have enough space to have, like we had 24. That meant only 8 per treatment, which is as much as we could possibly fit in our facilities. And so to get at some of those things, a much larger sample size would be really useful. And I don't know if Rachel Cook is on or not, but they also did some similar kind of things with elk, and they might have had a larger sample size and might have seen some kind of relationship there.
Yep. Other questions? Okay, so Michael Gant says, is a treated forest in the sense the same as a treated forest in wildfire terms, or is the connotation a bit different in this case?
Not exactly sure the question, but what I meant by treatment is either a harvest, basically. So for the herbicide ones, they were harvested, they were clear cut harvested, and then so that would be a treatment, an stand age treatment, and then we also had on top of that the treatment of the time since harvest, as well as whether they were sprayed or not. For the Northeast study area, the treatment was thinning. Basically, they call it fuel reduction, but I think it's simpler to think of as thinning. And they would go in and different levels of thinning. That's why we had 2 to 100% canopy cover. And then some sites we used weren't treated at all, meaning they hadn't had any harvest within 60 years, any thinning. Does that answer the question?
I think it does. Um, yes, he says it does. Okay, and Alicia Woods asks, do you know of anyone or lab that is doing microhistology of ungulate pellets to identify plant composition? I think that's a dying art, isn't it?
Yes, so Bruce Davitt at here at WSU was the go-to person for that kind of work until several years ago when he retired. And so a lot of people have asked that. I'd heard now and then that there might be people in Colorado doing it, but I think most of the people who do it are probably just doing it for their, they do it themselves and kind of train up on their own study area. But that's a really, really difficult skill. So even though I think it still holds its own even against DNA metabarcoding right now until we get some improvements on that. As far as I know, no one's doing it.
Okay. From Sebastian Botero wants to know, curious if you have data on how related are remotely sensed vegetation using NDVI to the ground measure of forage quality and quantity. And Aaron Wood also wants to know about that.
So I'll start with what we have actually done for the Northeast study area site. We did try to use LIDAR actually to try to get at forest biomass. So we have a little small paper that looked at that. And the relationship was not good at all until you had less than 50% canopy cover, and then it was reasonable. The problem with the LIDAR measures is that, at least at the time, it's really expensive to get airborne LIDAR, so you're only getting those once in a while. So they weren't necessarily very close to when the years that we were measuring, and in a different season. They were mostly in the fall where we sampled. As for NDVI, we have not done that well, let's see, trying to think. I think I might have messed around with it for this project. I don't remember the results. But I know that, for example, Ryan Long has used NDVI to kind of, he uses the suitable biomass, but then uses NDVI and some other things to kind of extend, to fill in the gaps because suitable biomass is measured on plots. So yes, I think NDVI is a little tricky for me to talk about mostly because the problem is that it kind of is doesn't measure biomass, doesn't measure quality, and there's a lot of problems with canopy cover. You can't get underneath. So I think in open grassland areas in Africa, there's been some work that kind of has strong relationships, but in the places that I work, I think it would be really hard to use. So that's kind of my answer.
Okay, and Kyle wants to know how your findings about forage quality in grazed and ungrazed compared to other related research, and if you would expect similar findings for elk.
Yeah, so I don't know if anyone who's used like the technique that we did by having animals measure their own habitat with grazed and ungrazed. But there have been, studies of wild animals on that. But I guess what I would say, there were elk in our study area. Um, there is probably depends a lot on how much overlap there is between diets, between elk and other cattle and other wild herbivores. So mule deer theoretically have less overlap between uh, cows because they're a small browser relative to a large grazer. So we might expect a larger effect of cattle grazing on elk. However, we restricted our animals to these grassland places. There were forbs, grasses, and subshrubs, but so we kind of forced them into an area where they would have to eat grass. And so I think if you allowed the deer to go wherever they wanted, they still do eat some, but they wouldn't eat as much. They actually ate about 60% of their diet in blue bunch wheatgrass, where we put them because they're required to be there. In real life, they would probably only eat a lot less. So I don't know about other studies that have measured it exactly like we do, but there are a number of studies that have looked at, animal movements and use of habitats for these animals in grazed and ungrazed landscapes.
Okay, and Tala asks, is it possible to measure diet overlap in a purely observational study?
I might need a little bit more information. Is this trying to get how to get diet composition? Yeah, maybe give me a little more there. I wasn't quite sure.
Okay.
Sorry.
Wow.
Or
Yeah,
just
go
say
ahead.
it, yeah.
Yeah, go ahead.
Yeah. Hi, Lisa. Thank you. That was wonderful. I was just wondering, we were looking at overlap between mule deer and livestock, uh, and thinking about any measure that we can take, look at direct observations of what the animals eat, or looking at fecal matter. It all is already a product of the competition, so it's not the
Right.
true overlap. And I'm just trying to figure out whether there's any way of looking at what would those animals, what would be the true overlap in the absence of competition
Mm.
when observing a natural system.
Wow, that's really cool. It seems like, and this isn't very practical, it seems like some large manipulative landscape level experiments with potentially with fencing, the fencing kind of thing can work, could be really hard at the scale you're wanting to work on and having wild animals coming and going in those. That's a really good question because wherever they are probably has past harvesting by those animals. And so, what happened 3 years ago could or may not affect where they're there. So I'm not sure I have any creative ideas at that, but it might take, a lot of money and some big experiments of exclosures to try to get at that.
Okay, and Mitch wants to know what your thoughts are on the extent of thinning. As you approach clear cutting, did you see any trade-off of increased forage availability relative to areas that were minimally thinned?
Yes, the thinning project was not clear cut. They were just removing a certain percent of the trees within a stand. And yes, we did have between huge difference in the amount of canopy and thinning that happened. And we did find a strong relationship. The more that was removed, the higher the suitable biomass that was available to them. So, for these animals, the forest can provide lots of important things for them, some thermal cover, some protection. But when it comes to food, even in terms of quality, most of the best food is going to be, at least where there's some openings to try to grow it. If you have a deep, dark forest, there's either nothing on the ground or a bunch of conifer, shrubs or evergreen shrubs that are not very nutritious and that animals tend to avoid. So yes,
I
the amount of
actually,
thinning did matter.
yeah.
Mhm.
I actually have a question. You showed one figure that on foraging time, and that you saw a strong relationship, but with the southeast was really driving it. Why do you think the southeast changed that foraging time so much?
Right. So I'll just start by saying, I think those people who've worked on like foraging behavior and these things before, it's really hard to predict foraging time. I don't think we have a really good handle. I threw a whole bunch of variables in there and we still really couldn't come up with it. So what I wanted to do is treat it equally to the others and show we use the same metrics to see what would happen. And if you combined all the data together, we had a relationship, at least was better than the null model. But when you look at it, like I said, it's not, doesn't seem to be a real relationship. It has to do with the fact that that Southeast study area. One thing could have happened on the Southeast study area is that those trials, most of the other two study areas, the animals were out in the field the entire summer. So they had to learn how to do this and do it well. And so that was our first project actually, and we kept them out for a couple weeks at a time. And so, they might not have spent as long time eating or potentially eaten as much because, they weren't forced to eat for so long of time on natural forages. So it might possibly be a design issue for that one. Um, it didn't seem to make a difference for the other relationships that we looked at, but it did seem like their time spent foraging was shorter. It also could have been, both of those, they all happened, foraging time was measured by an activity sensor that was then calibrated with observations. But calibration can be tough. And although our calibrations were about 80% accurate, it's possible that it's pretty hard to measure foraging time per day. And it's possibly there could be something different about how that happened on that project.
Okay, great. And Dan would like to know if when you're using the fresh model to estimate the suitable biomass, is there an R package available for that?
Um,
Not a package, right?
there's nothing published that I know. I do have an R package that I can share. This is actually something that Don Spallinger made. He and Tom Hanley and a computer person, I can't remember his name, developed the first fresh model, and it was a web-based one where you had to enter all the data in. And we used it for some of the first works that we did. However, that's extremely annoying. And so I asked him if he would make an R package, and he did for it. And he's has several versions that he's worked on, but we have used that, and we have shared that through our what is it called? Our workshop, nutritional ecology workshop, and I think could be shared otherwise. I mean, if you're good at R, you might have to do your own tweaking to make it kind of work for you, but yes. So
Yes, Lisa
let me
has
know.
taught one of my students to use it in R when we were working on our project. So she just zoomed in with one of my students and was gracious enough to explain how to do that in R so she didn't have to enter all the data individually.
Yeah.
So it is possible. All right. Any other last second questions? All right. Well, thank you, Lisa. It was a pleasure to be able to reconnect with you over Zoom and see some of the work that I was able to see in action over the years, summarized like that. And if anyone has suggestions for our 25-26 webinar series, or you would like to be added to our email list, please send us an email at that site that is listed on the screen there, or email address. And you can also check out our past recordings on our website as well. And so thank you for closing us out, Lisa, for a great year of our webinar series.
Thank you.
Thank you, Dr. Shipley. Appreciate it.